Actually, I got the Nobel Prize for my work on photosynthesis, and as such, I'm quite often asked whether we can use plants to produce biofuels to solve the energy problem of mankind and to reduce global warming. So we have to understand the limitations of photosynthesis, its basic science, and try to improve the efficiency of photosynthesis as applied science. Some basic facts about photosynthesis. We have the light reactions, where absorption of light leads to the creation of chemical energy and oxygen. And we have the dark reactions, where the stored redox energy is used to take carbon dioxide out of the air and convert it into sugar. Quantum yield in photosynthesis is close to 100%, but this doesn't mean that each photon absorbed leads to an electron transfer across the membrane, but the energy yield in general is low. Plants can only use 47% of the sunlight, and depending on the wavelength, between 19 and 33% of the energy of the absorbed photon can be stored in the form of NADPH, which is fixed hydrogen. Then we have further reduction by inhibition of photosynthesis, damage at high light intensities, and inefficiency of carbon dioxide fixation. The efficiency of the conversion of sunlight in photosynthesis depends also on the color of the light of the absorbed photon, and at the end we can use about 19 to 33% of the energy of the absorbed photons in the form of NADPH. And in the scheme here in the middle you have some problem, because the complex which is seen in the middle actually is supposed to provide the electron acceptor for the photosystem too, and there is some limitation. So we have for the inhibition at high light intensities, and which actually means that photosynthesis is saturated already at 15% of the full sunlight. So 85% of the full sunlight cannot be used for photosynthesis. And another point is that we have the enzyme Rubisco. Rubisco takes the carbon dioxide out of the atmosphere and incorporates it into a sugar called ribulose 1,5-biphosphate. And this enzyme is a very sluggish enzyme, and actually, it's one of the slowest enzymes which is known, and it discriminates poorly between carbon dioxide and oxygen. And in every 35th reaction cycle, incorporates an oxygen instead of a carbon dioxide. This leads to an incorrect substrate, 2-phosphoglycolate, which has to be degraded by a lot of energy by the plant. Then at the end, we store about 1% of the sunlight energy in the form of biomass, and when we convert this into biofuels and biogas, we get even much lower values. So in German biodiesel, the most negative example, there's about 0.08% of the sun's energy is stored in the biodiesel. Brazilian bioethanol, 0.3%, palm oil, diesel is 0.4%. In biogas, it's about 0.2%. Of course, in the conclusions then are, if you use photovoltaic cells, electric engines and batteries instead of biofuels, combustion engines, we use the energy of the available sunlight by a factor of at least 500 better than we would produce biofuels. If we use biogas to generate electricity, we are by a factor of 100 less efficient than by using photovoltaic cells. Here are some numbers concerning the German biodiesel from rapeseed. And 10% of Germany's cropland is used to grow rape. The annual yield is about 4 million metric tons. Additionally, 5 million tons are imported. And these 9 million tons are used to produce 3 million tons of biodiesel. And this number would be sufficient to replace 9% of Germany's annual diesel consumption. Thus, when using all the cropland of Germany for biodiesel production, this biodiesel would just be sufficient to replace 40% of the diesel consumed in Germany. So we have to improve photosynthesis, and what has been achieved. There are alternative routes for degradation of phosphoglycolate degradation, and there's an increase of up to 50% in biomass. We can optimize the Kelvin cycle, gives another 60% increase in biomass. We can increase the B6F complex, which I mentioned, which gives up to 71% increase in biomass. And these improvements appear to be additive, and there's a good chance to double or even triple the efficiency of photosynthesis, leading to a corresponding increase of biomass and group yield. This works so far for model plants, Arabidopsis and tobacco, but we have to actually use the knowledge from model plants like Arabidopsis or tobacco to improve the yield of wheat, rice, and other crops by genetic engineering. But we have to convince the public that this procedure remains the best way to ensure that there is enough food for the still increasing world population without the need to further deforestation and reduction of other natural habitats. And an improvement of photosynthesis also would reduce global warming. Of course, the alternative to biofuels would be direct electrification via electric technologies. And so we have to stay electric. We have to transfer electron. And if we use the high voltage direct current, we lose about 3% per 1,000 kilometers. And there's a new cable planned from Australia to Singapore, which I think is an extremely positive development. We can store energy in batteries. We can use batteries and electric engines to drive cars and use heat pumps for heating buildings. And there's the advantage, there's nearly no loss of primary energy. And therefore, my take-home messages are, do not use agricultural land to produce biofuels, improve photosynthesis for food production, and stay electric as long as possible, use batteries for energy storage, and use cables for energy transfer. Produce hydrogen where required by water electrolysis and by methane pyrolysis, and introduce carbon pricing worldwide. Thank you very much.
Thank you. Thank you very much, Professor Michel. Next up, we have Marc Fontecave, the Eni Award Laureate for 2024.
Yeah, thank you. I have waiting for the slides too. Oh, yeah, thank you to the organizer for allowing me to tell a few words about what I think is one of the most fascinating and in contrast to what I hear sometimes, the most urgent challenge for humanity, which is successfully transitioning to a world with much less or with no fossil fuels. In fact, we are too far from it, and because we have to deal with, we have no miraculous solution. We have to deal with solutions which all have drawbacks, and we really need to massively invest in research and innovation in the field of new energy technology. I feel legitimate to say these words, in particular as being the president of the Energy Committee of the French Academy of Sciences since 2018. The Academy has made a huge work on this issue. Yeah, to summarize, I mean, the energy transition implies three directions. I mean, the first one is to decrease the energy demand. Of course, it will not be the same extent in all countries. I mean, some will increase it and some can decrease it. The question is indeed to which extent, and they work on the energy efficiency technology. The second thing is we need to massively electrify the society. Again, to which extent the share of electricity can be in different countries, this is open. But it means not only consume more electricity, but at the same time produce more low-carbon electricity, I mean, nuclear, solar, wind, and so on. And on the right, you see the kind of trend for the next 30 years, where the aim is to increase the blue part, increase the, I forgot, sorry, the third point. This is very important because the world will not be 100% electrified. And I must say that the cry, decarbonize the planet, is quite inappropriate because the planet is very carbonized and will be very carbonized in the future, and we will need alternative sources of non-electrical carbon and heat, and biomass will be part of it. We have heard critics about it, but for the moment it's mainly wood, and as you can see, the gray part will increase, but to a minor extent. So what we have done best so far regards the production of electricity supply. And this is through the development of massive capacity of solar and wind plants, together with promoting a new renewal of nuclear energy. In contrast, I would say that we have been less successful, and we need research and innovation in electricity storage, which is absolutely requested for dealing with an increasing share of intermittent sources of energy, I mean solar and wind. And by storage, I mean massive capacity of electrochemical storage, like batteries, mega batteries, chemical storage with hydrogen in particular, and hydraulic storage with more pumped storage power stations. We have not either succeeded in many parts of the world to stimulate the electricity demand. So we increase the electricity supply, but the electricity demand is not coming at the right level, which means electrifying devices in housing, in transport, of course, electrical vehicles, electrofuels, and also making industry more electrical with new electrified process for making steel, cement, concrete, glass, ethylene, hydrogen, and also for introducing electrical heat massively into the industry. And Europe is particularly affected by that. As you can see in all these curves, in the last 10 years, the demand of electricity has not increased, but has decreased. So we are not electrifying our society in Europe. And in contrast, in the world, it's about plus 4% every year. Yeah, so there are, in fact, things are not going too fast because there are so many technical problems. So we really need more basic technological industrial research altogether. I mean, this is a list of different domains where research is still needed. A lot of problems have not been solved, in contrast to what people say in general. I mean, like producing green hydrogen, technologies for recovery and recycling metals, plastic batteries, improving solar cells, improving batteries, decarbonate the industry, new processes for biomass valorization, new nuclear reactors, small modular reactors, and fourth generation reactors, and so on. And I want to finish on the last point, on the green organic chemistry and carbon capture and utilization. That's the kind of things we are doing in my lab, where we try to do some kind of artificial photosynthesis in which we combine solar electricity to electrolyzer to convert CO2 that has been trapped somewhere to make hydrocarbons, alcohols, fuels, plastics, and for example, sustainable aviation fuels, which will be absolutely needed because there is no change that big planes will use hydrogen or batteries, I think. Yeah, on that, I conclude that, again, it takes time to make this transition, and it will mostly depend on progresses on all the directions that I've indicated. Thank you.
Thank you very much. Thanks for that. Next up we have Professor David Baulcombe, who is the Lasker Award recipient for 2008.
Thank you. Opportunity to talk to you today about the science that we've been doing. If I could have the slides, please. What I'd like to do is to make the case for curiosity-driven science. Funding agencies, governments, philanthropists like to justify the resources that they allocate to science by an alignment with the challenges for humankind. Now, I'm not saying that we should ignore these challenges because they will not solve themselves, But what I am saying is that if we are to meet those challenges, we need to supplement our strategic science with an ever better understanding of the natural world. We need to know, in particular, the things that we don't know that we know. So the good news is that one approach to doing this does not require you to be a genius. All it needs is for you to have the intellectual agility to notice the results that you get that don't make sense. I would like to illustrate this point using an example of some of the work that we've been doing over the years. So my interest was in genes. I wanted to know how genes are switched on and how they're switched off. And I decided as an experimental system I'd work with viruses. And one approach we took was to take genes out of viruses and we put them into plants using transgenic technology. The expectation was that these genes would be active, that they would produce proteins, and that those proteins would play a role in the functioning of genes. Actually, what happened was that that didn't happen. The gene was not expressed, it did not produce protein, and rather than promoting the replication of the viruses that we inoculated to the plant, it actually inhibited them. What it turns out that we had done was to activate a pathway in plants that resulted in the production of small RNA molecules. And these small RNA molecules served as the specificity determinant for effector proteins that block the expression of genes. So this was an unexpected result. It took us into the understanding of a pathway that is important not only in plants, but also in other organisms as well. So why is this important? Well, firstly, it turns out that the system that we had activated in the plants using this transgene is a natural immune system that operates in plants that protects plants against viruses. It explained a lot of data that were in the literature going back to the 1920s. It explained a lot of things that we knew about the behavior of plant viruses that we had not otherwise understood. It also told us new things about RNA and what it does in plant cells. So on the left-hand panel here, we've got an illustration of an RNA molecule that moves between cells. We didn't know that RNA could move between cells previously. It also told us about a link between RNA and what happens at the chromatin and DNA level. So it shed light on mechanisms associated with non-Mendelian inheritance, epigenetics. There's a very interesting phenomenon known as paramutation that our work took us into. So new findings about RNA, epigenetics, and chromatin. So how can we use this? Well, we can harness it to produce plants that are resistant against virus disease, and our initial experiments showed us how to do that. It's a very simple experiment. You just insert a transgene that corresponds to the viral genome, and you have plants that are resistant against viruses. This is a very versatile technology, and it could be used to protect almost any crop against any viruses. And one of the few frustrations in my career is that due to global resistance to GM technology, we have not been able to deploy this on a large scale. This is one of the examples from our work. The work that we've done in my lab is all on plants, but as Darwin showed us, we're all part of one tree of life. And so things that you find out in one organism may well be true in others. And so it turns out with RNA silencing. So we could, for example, use essentially the same approach that we use to protect plants against virus disease to protect farm animals against virus disease. And again, due to the resistance to GM, this is a really badly needed technology that could protect the planet against the next pandemic that has not yet been used. You can also, and this is not our work, but work of a company called Alnylam. They've developed small RNAs as drugs, and protect the people against various diseases. So these aren't just the only examples of where unexpected results have shed light and benefited human beings, and these are just a few examples that I was able to get using AI from the literature, but there are many more that you probably all know about. So I've greatly enjoyed over the years working with very talented people, with funding agencies and philanthropists who've been sympathetic to the idea that curiosity-driven research is important, and I hope that you too will have the opportunity to do that. Thank you.